This is episode 2 of Sam and Tiv Talk Science.
Yes.
What are we going to talk about today, Sam?
Okay, so we're going to talk about so it's the 5th of April, and we're going to talk about the 3 biggest breakthroughs, the 3 most important science breakthroughs, medical breakthroughs from March.
Exciting. So I think I'm going to let you kick it off. So what discovery do you want to start us off with?
So, okay. So we're going to start by talking about a drug called Lenacapavir.
Lenacapavir,
which I think I'm saying it right, which is one of my favorite medications. And if you follow me on Twitter, you see I'm a little bit obsessed with Lenacapavir.
All right. For those of us not on Twitter, tell us about it. What kind of drug is it? So
it's by far the best preventative measure we have for HIV that exists, full stop. It prevents HIV. So what it is, it's an antiviral, an antiretroviral, which kills HIV. And it's intended as pre-exposure prophylaxis, PrEP.
And what does that mean, prophylaxis?
So you give it 2 times per year. And if you give it 2 times per year, then it stops you from getting HIV. So it's not something you take if you've got HIV, you take it before HIV. So not necessarily before getting specifically exposed to HIV, but you just take it twice a year and it stops you from getting HIV.
So that if you do get exposed, you don't end up getting infected.
Exactly, yeah. So it blocks the replication of the virus. So twice a year, and it's an antiviral that sticks around in your system for the whole year. And if you're exposed to the virus, HIV, then it just kills it.
Wow. And why is it such a big deal compared to, existing HIV drugs?
Exactly. So the reason it's such a big deal is because last year, so we're talking about this from March, because there's an update, but last year there was the big phase 3 that was published, looked at Lenacapavir for HIV prevention, and my favorite statistic, the efficacy was 100%.
That is insane.
So 100% efficacy. So it's very, 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 very rare that we get a trial or medication that has 100% efficacy, and this is one of them. That's why I'm so excited about it.
Wow. And how does this compare to what was previously available?
Exactly. So the other alternative, essentially, is PrEP pills, which lots of people take. So pre-exposure prophylaxis pills, which you take, so in the trial, which is based in sub-Saharan Africa, they take them every day.
Every day?
Yes. There's an alternative. So if you're in the UK, let's say, you can use PrEP when you think you might be exposed to HIV. So if you're going to have sex, then you can take PrEP, so for a few days before and for a few days after. And that's a very effective way to prevent HIV. Now, but the issue is with adherence. So if it's a pill that
you have to take it every day.
Exactly. So if you have to take the pill every day, then adherence is an issue. So if you do manage to stick to 100% adherence, then it will work for sure. It's very effective. But sticking to that is very hard for a few reasons. 1, because you have to remember. 2, because it causes side effects.
It does. What are some of the side effects of the pills?
Main side effects are things like nausea, vomiting, diarrhea, which is just not very nice to have to deal with. Right?
And yet you're taking this pill every day. That could cause these side effects.
Exactly. And so, of course, people don't like to take the pills every day. If you don't take it every day, then it drops the effectiveness of the drug. So with Lenacapavir, you don't take it once every day. You take it twice a year. And even if you take it twice a year, so it's an injection, I should say that.
Yeah.
Subcutaneous injection underneath the skin. It was 100% effective. And that's basically really because it boosts adherence.
That's incredible. I mean, going from taking something every day to just 2 injections a year.
Exactly.
And is it affordable, you think, as well?
So no, no, but that's so at the moment it is a course of Lenacapavir at the moment is tens of thousands of pounds, and I know that's ridiculous, of course, but with scaling it up and with maybe some deals that are done, there's studies that have shown that we can get the price down from tens of thousands of dollars to under 100 for a year.
That'll be amazing.
So at the moment, is it affordable? No.
But it's getting there. It's getting there.
And so the update to Lenacapavir from March 2025 was, so what's better than 2 shots per year or 1 shot a year? So
Did they manage to get the same results?
yeah, so basically yes, with some caveats. So they made so it's a study from Gilead Sciences in the Lancet in March, and they
Huge journal.
exactly,
Yeah.
and they reformulated it so that it's rather than subcutaneous, it's intramuscular, and they edited it in some other ways.
Yeah.
Which basically means that you can give it just once per year rather than twice.
So
per
then
year.
it will last longer in the body, and you don't have to have 2 injections.
Exactly that. So now we have to say the caveats. So the study in the Lancet that we're talking about is a phase 1 study.
Yeah.
So they're just looking at safety and also levels of the drug.
But they have already finished the phase 3 study for the two injection.
Exactly. And so what they did, really the nice data from the study is that if you look at the levels of lenacapavir achieved by twice a year dosing over the course of the year with 100% efficacy, it's like this, let's say. And with once a year dosing, it's the same or even higher dose throughout the year.
How did they manage that? Was it from the formulation or doing the muscular injections?
Exactly. And suggesting, so although there is no efficacy data, it's suggesting that if you can achieve the same dose or even higher, then efficacy probably is going to be the same.
Wow, that's incredible. I mean, that's going to cut costs in half, and like adherence is going to be even easier.
Exactly. So exactly that. Reduce cost, make adherence easier. It makes it easier for patients if they only have to go once a year. It makes it easier for the staff because if they only have to do it once a year, it just makes it easier.
That is incredible. And I have some, as a scientist, I've got some, more biochemistry questions for you. So
biochemistry? I'm not going to...
Oh no, the degree that you did as an undergrad. All right, you studied
I did.
You did.
I mean, I'm not going to... Okay, go ahead.
Yeah, but, just high level, how does this drug work? How does it stop HIV from, replicating or infecting?
So it stops the replication. It stops the infection by inhibiting it's a capsid inhibitor.
I see.
So
okay.
I have to say I don't really know exactly how that works.
Oh no, that's all I want to know. I just wanted to know like how it interacts with the virus.
Exactly.
Yeah.
So it's a capsid inhibitor, and that stops the virus from replicating.
And
that's
apparently
as far as I.
It's first in class, so it's the first time a drug like this has been made for HIV.
Exactly, and I should say that this lenacapavir in the twice yearly formulation is not FDA approved, but it's now had its two phase 3 trials, and so we're expecting approval in roughly summer of this year.
Amazing. And is this the video you shared on Twitter where there was like a standing ovation when the results were announced at a conference?
That's right. So it's not just me that's excited about this. People love it. 100% efficacy. How can you not love that? No.
How many times do you see that in your lifetime?
There's another example. I actually went out of my way to look for all of the examples where we have 100% efficacy in a.
Of course you did.
And there is, so some of the vaccines can do it. I think, if I'm not wrong, some of the vaccines can do it.
Yeah.
But
you did mention, I don't know, HPV vaccines.
Right?
Yeah.
So for prevention of cervical cancer.
Yeah, exactly.
Yeah, so I don't think that's from a randomized setting, but still, like if you look in like the cohorts of people, there's a cohort of people in Scotland, for example, who have had the HPV vaccine, the incidence of cervical cancer is 0.
That's amazing. I mean, it's just like, what a time to be alive to, witness all these things having 100% efficacy.
Yeah. There is another one, which is the GLP-1. Had 100% efficacy in the trial. But that's a different story.
That's another rabbit hole we can go down.
Yeah.
Wow, that's amazing. So can you say that we've like, I don't know, if it's done in a prophylactic setting, like HIV is basically curable, right? If you give it out to everyone, like who might get exposed to it, then you can technically cure it from now on.
Yeah, it's not a cure though, right? Like an absolute prevention. So you don't even get it in the first place.
Yeah.
But in terms of like eradicating HIV from the world, I mean, it's an extremely ambitious vision to say that you might be able to eradicate HIV, but
technically, if
why not?
we all, took that.
Yeah. So I mean, in countries like the UK, where clearly our HIV burden is lower, it's still there, but it's lower. We still have HIV, and we can drive down HIV to eradicatable levels within our lifetimes.
That is amazing.
In terms of around the world, clearly it's trickier.
Yeah.
But this is going to help, and certainly, even if it doesn't allow us to eradicate HIV, it certainly will drive down the levels.
This is amazing news. I can see why it's in the Lancet.
Yeah, and this is I love this drug. Any news on this drug is news that I love.
Wow, awesome.
Any other questions on that one? We can move on to the next one.
I think I've asked most of the questions.
Yeah, I think
is there anything else you want to bring up?
I don't think so. I think we covered it well. I think.
Awesome.
So the next one?
I think the next, being your resident cancer vaccine geek, I guess I have another cancer vaccine update. So this study was published recently, maybe not in March, but still relatively recently. But it's a very exciting study in Nature in kidney cancer, high-risk kidney cancer, where it's personalized cancer vaccines in 9 patients for renal cell carcinoma. And the reason this study got such a lot of press is that all 9 patients were free of recurrence after 3 years follow-up, which is huge, right? Like in the clinical space.
So they have an awesome figure about this. Have you seen the figure?
Yeah.
They have an amazing figure where so if you look at the historical recurrence rate so this is for stage 3 or 4 renal cell carcinoma, stage 3 or 4 kidney cancer. The recurrence rate at 3 years, the expected recurrence rate at 3 years would be about 50%. So you would expect of these 9 patients, you'd expect 4 or 5 of them to have a recurrence of their kidney cancer.
But none of them did.
And the number was 0. So technically, so there are clear caveats to these data, but technically the efficacy in the trial was 100%.
Yeah, and it's
with caveats.
And yeah, with obviously like small control. I'm sorry, no controls and a very small population.
Yes,
but still, fingers crossed for their phase 2.
Yes.
But this is a huge deal because, RCC is a cancer type that's known for having a low tumor mutational burden, unlike melanoma, where it's easier for immunotherapies to work. So the fact that they got such an amazing result from this personalized cancer vaccines is opening up doors for other cancer types with a low tumor mutational burden.
Yeah, exactly. And so tell us about the patient journey for someone getting this vaccine.
Yeah, so, this was a phase 1 study, but what they did was they did RNA sequencing on each of the patient's tumor to find out which mutations they have, and they also did whole exome sequencing. This is on the DNA level. And then they made these vaccines. It takes several weeks, usually 8 to 10 weeks, for personalized vaccines to get made after the resection of the tumor. And they put up to 20 neoantigens specific to the patient in the vaccine and then delivered it to the patient. And obviously, some of them had checkpoint inhibitors as well, if they were eligible, I believe. And there was also an adjuvant being administered, and that was just to kick up the immune system to get the immune cells going. But yeah, and this was a long follow up, but the results are really promising.
Yeah, 100%. Yeah. So after surgery, right? So last week, or previously, we talked about your guys's. So we should say that you have. So you said at the start, but you have you're part of a cancer vaccine company too.
Yeah.
And we mentioned that one of your trials you're doing for esophageal cancer. You're doing it for the vaccine before surgery, right? But this is the vaccine after surgery. So you take the tumor out, you sequence, and then you give the vaccine, and then it prevents recurrence.
Exactly. So this is a good point of comparison because for the company I'm working for, Infinitope's Cancer Vaccines, we're doing semi-personalized off-the-shelf vaccines, which means you can give it before the patient goes through surgery to have their tumor resected. You can inject them a few weeks before the surgery with the vaccine to kind of prime your immune system to get ready to soak up any remaining cancer cells. But here in this study, because it's a personalized setting, and it's not just this study, it's any company trying to make a personalized cancer vaccines, you need the patient's tumor to so you need to wait until the surgery and the resection to get a piece of that tumor to then do the DNA and RNA sequencing to figure out which mutations are there and then create the cancer vaccines for them. So, there's a disadvantage of, not having the vaccine earlier, which we think is always better because the later the patient gets the cancer vaccines is when they're more likely to be more immunocompromised. And, patients are usually stronger the earlier on in their cancer progression.
Interesting.
Yeah.
And so technically, these guys are competition for you. Is that fair to say?
I think it depends on, from an industry point of view, it depends on the cancer type. Because everyone's
if you're not targeting the same cancers, then it's not quite.
Yeah, then it's not a direct competition.
Yeah.
And we could talk about pan-cancer vaccines, but that's still,
Wow.
these are still
vaccines?
That would be amazing. So some companies are exploring that space.
That's crazy.
Yeah, like they would look at, for example, KRAS mutations, very common in 30% of cancers or something like that. So if someone can make a KRAS vaccine and give it to all cancer types affected by KRAS, then that'll be amazing.
And that's like super off the shelf, right? That's even more that's like the highest level of off the shelf. That's just like the most, I guess, like simple version of the
Exactly. But caveats, obviously, HLA type. HLA is the context in which whether or not that vaccine is going to work.
And resistance to, I assume that if you had a vaccine that only targeted one neoantigen, then the chance of resistance is just higher.
Yeah.
Is that fair to say?
And it's the same logic as with all the KRAS small molecule or inhibitors.
Exactly. Yeah. You have to hit more than one pathway at the same time, really.
Exactly. That's the issue with going for something so, it's a huge driver mutation, but it's also, the tumor is going to fight back with like these resistance mechanisms.
Yeah, exactly.
Yeah.
So, okay. But that's interesting. And we had a bit of a critique, a bit of a criticism of this paper, right? Or of the one of the findings.
Yes, we did. So, they after the vaccine was administered, the scientists obviously measured the immune cell profile of the patients by getting their blood, and it turns out most of the immune cells that were stimulated were CD4 T cells, which are not the actual immune cells doing the killing of the cancer cells. They're more like the, how do you say this? The admirals in an army. So they don't really...
They direct the response, but they're not doing the killing at the front line.
Exactly. Thanks.
Right.
Exactly that.
Yeah, interesting.
Yeah. So, but still, obviously, like no recurrence is still a great result, but usually people would like to see more of a CD8 T cell response, and CD8s are the ones that do the cytotoxic killing of the tumor cells.
So the CD4 T helper cells are clearly seem to be doing something. They seem to be making sure the cancer doesn't come back. But really, what we really want to be seeing, and what you guys are looking for when you do your studies, is the killer CD8 T cells, not the T helper cells, which are good, but they're not the best.
Yeah, you really ideally want, I'm not an immunologist, but from what I can see from immunologists, they would like to see a CD8 T cell response. But I mean, encourage to give this, paper credit. They did look at the CD8 T cells, and they did something called TCR sequencing, which is you take the receptor on those expanded T cell clones and you sequence it. And they found that all the patients had at least TCRs or T cells with specific TCRs to the neoantigens, half the neoantigens. So half the neoantigens, expanded T cell clones specific to them. So that's pretty promising.
That is promising. Yeah. Quick tangent. I think is it fair to say that making a cancer vaccine which has a very strong CD8 T killer cell response is hard? Is that?
So I think you would have to be a really good vaccinologist and immunologist in order to create that sort of response. And at least I can say in infinitopes, I can't say what we're doing, but we're able to achieve that,
and
which is like,
so
the gold
you can
standard.
add adjuvants, I guess. You can add things to your vaccine to try somehow to stimulate that T killer response. Is that?
Absolutely. And I mean, lots of people use like PD-1 or PD-L1 inhibition to really unleash the immune system onto your cancer cells. But, that comes with its own problems with toxicity. And, there are some cancer vaccine trials where if you don't give the checkpoint inhibitor with the vaccine, then the effect is gone. So it means that you're really relying on the checkpoint inhibitors and the real effect is not coming from the vaccine.
There was this beautiful, innovative study that I saw last week where what they did, and you can tell me what you thought of this. What they did essentially is they built the antigen, the tumor antigen, and a sting agonist. So an agonist of one of these immune proteins. Yeah. They built the antigen and the sting agonist into the same molecule. And it turns out if you do that, then you produce a way better CD8 T killer cell response. Because essentially what's happening is that sting is taking the antigen to the ER, to the endoplasmic reticulum, where it gets chopped up and presented better on the surface of the cell.
Ah, I see.
So the antigens, in other words, are presented better, and that activates T killer cells better.
That's amazing. So it's basically encouraging the antigen presentation pathway.
Exactly. Because the antigen is going to traffic to the ER where it's getting chopped up. Yeah. And the other thing is that, of course, in those same cells where the antigen is getting chopped, you activate sting. And one of sting's functions, it turns out, not that I knew this, is to activate T killer cells. Wow. So that was a preclinical study, but that would be one way to activate the T killers, I guess.
Yeah, it's one way of adjuvant.
Yeah, I mean that's so
creative.
It's super creative, right? I mean, it took me a little while to understand that, but when I did, that's crazy.
And, science, biology just surprises you every day.
It does. It really does. It's so interesting. Okay.
But yeah, that was very encouraging for cancer vaccines. And I guess my only last comment would be, it'll be nice if the study says that RCC, even though it has a fewer number of mutations, so a low tumor mutational burden, it has a lot of well-defined driver mutations, such as in the VHL, I believe. So that makes me think, like, why can't they make an off-the-shelf vaccine if these driver mutations are well known?
And just to make it super clear what the benefits of the off-the-shelf vaccine are, it's cheaper, faster. Mm-hmm. Anything else? Or is those the main ones? It's just faster and it's cheaper and that's better.
Yes, you can give it earlier on in the treatment regimen, and it's cheaper, which is a huge deal in healthcare economics. And
not to downplay the importance of price.
Yeah, exactly. So I guess that would be my only question to the authors of the study, but I guess their response might be that there are different mutations amongst those genes that get mutated. So but I mean, fingers crossed for phase 2 and really great news for the cancer vaccines field.
For sure. Super interesting. Okay.
All right. And Sam, you mentioned, you mentioned the ER and the proteosomes. So I think that's a good segue into our last and third study of March 2025.
I've got it in front of me.
Oh, yeah. He's got, for those not watching the video, he's got the actual Nature journal subscription, which I don't see much of nowadays. So it's so great that you do that, honestly.
I just love it too much. I can't help it.
Tell me, what's the third study of the day?
This is the discovery that, it's in Nature from March, and it's from the Weizmann Institute of Science in Israel, in Nature. So, and it's the discovery that our cells, as we speak, are producing their own antibiotics.
That's crazy. How exactly?
So let me just say, so clearly the world is facing a big problem of antibiotic resistance, right?
Yeah. Describe that problem to us a little bit for the audience who's not familiar.
So we are facing bacteria which are which are infecting people, which are causing sepsis, so causing overwhelming infection, affecting organs, which are resistant. They don't die when we give them lots of our antibiotics. So, for example, penicillins. So we give a penicillin, and the bacteria are resistant, and that's a big problem, and it's causing.
And it's because they're evolving, and
yeah, exactly. So they evolved by Darwinian natural selection.
Yeah, and it's also because in some places clinicians tend to
that's right
prescribe antibiotics even though there's no real need for it.
That's right. So things like viruses. So at med school they spend a lot of time making sure that you're not prescribing antibiotics for virus infections. There's lots of schools. So for example, if you have a sore throat, then you want to make sure that sore throat is caused by bacteria, not a virus, before you give antibiotics.
But usually you don't have enough time to discern that, so you just give the antibiotics anyways.
Well, we should. We definitely should be resisting the urge to give antibiotics, and there are good ways to do that.
Yeah.
But for sure, it's a big problem, and it's getting worse. And to be honest, it's killing millions of people a year.
Oh God.
And getting worse.
Yeah.
So we need to solve the problem. One of the ways you can solve it is to make or discover new antibiotics.
And how does this paper kind of shake this up?
So it turns out, the proteasome, it's a big enzyme in our bodies which chops up proteins that we don't need anymore.
And like the understanding has always been that it's just to recycle the proteins in your body, right? And to break it down and reuse the broken down bits for making future proteins.
Exactly. It's like a recycling bin.
Yeah.
And the peptides that it produces, so peptides are broken down proteins, the peptides it produces, like you say, basically don't really have any function.
Yeah.
Well, no, it turns out they do, and they're antibiotics. So constantly,
that's huge.
Constantly, the proteasome is just going all the time, right?
Yeah.
And constantly, it's producing these peptides just from regular old proteins. Any old protein, essentially, can
just a normal proteins in your cells.
Anything. It's not a special protein. It's not an immune protein. It's nothing like that.
Yeah.
Just any old protein. It chops it up, and it produces a little peptide.
Yeah.
That can kill antibiotics. So that's happening all the cells, all the time. But then, if you get an infection with the bacteria, then it ramps it up massively. And then you produce even more antibiotic peptides to help you stave off the infection.
This is crazy. This is paradigm shifting.
There's a great line in the study. So do you want to have a guess at how many new potential antibiotics are discovered here?
Oh gosh, I forgot. 300.
The number is a bit ridiculous. The number is 270,872.
Oh my goodness.
So this, which is an insane number, right? Of potential antibiotic peptides are discovered.
How do they define it as a potential antibiotic peptide?
So it has to have a precise sequence. So they did it's technical stuff, in silico screening of these sequences that they have discovered that can kill antibiotics. So they validated experimentally 10 of these 270,000, and all of them do kill antibiotics, do kill bacteria.
Yeah.
And the most exciting part of the study, I think, is that they use one of these antibiotic peptides, which just comes from a regular old boring old protein, in a mouse model of Pseudomonas aeruginosa. Which is
a long word.
It is. So Pseudomonas aeruginosa is a bacteria which is notoriously resistant to antibiotics. And in their mouse model, if it's a mouse model of sepsis, so overwhelming bacterial infection induced by Pseudomonas aeruginosa, and in the mouse model, if they give this new antibiotic they've discovered to the mice, not only does it kill the Pseudomonas aeruginosa, but it stops the mice dying from sepsis.
Wow.
So
that's insane. And this is one of the 10 that they found. And they see that the sequence should be able to be an antibiotic specific to the pseudo bacteria.
So not just pseudo, yes, but lots of different bacteria, but one of which is Pseudomonas aeruginosa. And so this is a bacteria which is resistant to lots of our currently used antibiotics, but it's not resistant to this one.
That is incredible.
Yeah. So
wow,
super cool.
And it's been right under our noses this whole time.
Literally,
So our bodies are churning out antibiotics. Do you think this is why we're not infected as often as we should? Like our bodies are already churning out these antibiotics. So
very, very interestingly, one of the proteins they discover that's part of this pathway, this antibiotic pathway, people who have mutation in that protein are immunosuppressed.
Wow.
So yes, in other words.
So is this something in the proteasome? Is this gene in the proteasome?
So this gets into the real details, but essentially what they found is that if you get an infection with a bacteria, then the proteasome gets modified by this protein.
Yeah.
Gets modified, and that just makes it churn out more antibiotic peptides.
Wow.
But if you have a mutation in this protein, you are immunosuppressed.
That goes to show proteasome is part of the immune response. I mean, we know about the immunoproteasome where your proteins get chopped up and then presented on the surface of your cell by MHC, but this is different. This is making antibiotics.
Exactly. Yeah. So the other thing is this is so cool is that we use this in the lab, right? This thing called bortezomib, which inhibits the proteasome. You must have used it.
Yeah,
right?
yeah.
Use it once or twice.
Yeah.
So, but it turns out that if you use bortezomib, then you are much more sensitive to bacteria.
Wow.
So you block.
Because you're blocking the antibiotic producing pathway.
Exactly.
That is mad. And I think there's somewhere in the paper that was saying that like one of the proteins that get chopped up by the proteasome, and that becomes antibiotic peptide, is also a component of the proteasome, which was like a bit meta.
That's crazy. Which one was I didn't see that, but that's crazy. Yeah.
I just remember coming across it somehow, but yeah, that's really meta. So these proteins have nothing to do. Sometimes they have nothing to do with the immune system. They're just a boring old protein.
Okay, so the top hit, right, that they discovered, what it does is a serine threonine phosphatase. So it's like the most boring. No offense to the phosphatase lovers, but it's like the most boring of all proteins. Doesn't do. Okay, it probably does some things, but it basically doesn't do anything. But if you chop it up, it can kill antibiotics. Yeah, it can kill bacteria. I mean,
this is nature being super resourceful, and
it's wild, right? But I love the idea that we're sitting here, like, strengthening ourselves with antibiotic peptides.
Oh my gosh! So are they gonna try? So I guess my question is, if our body's already producing these antibiotic peptides, why are we still not able to kill some of these bacteria that's causing like sepsis, for example? Is it because we're not producing them to a high enough amounts and we just need a little kick?
That's right. Yeah, I think that's exactly it. Well, I guess it's complicated. It's a good question, but I guess it's because exactly that. So it's just not right. It's not sufficient. So, but if you synthetically, and they proved this, if you synthetically produce it and then give it at a proper pharmacological pharmaceutical dose, then it does work. So clearly the peptides are killing antibiotics and are working to resist infection.
Yeah.
But clearly, obviously, it doesn't work all of the time, and people still do get infections and still get.
It might be a numbers game. So maybe if they have the synthetic version being administered, then that could be a potential. Yeah,
exactly. And I guess this is, to be honest, this is a totally new pathway, right? So
it's paradigm shifting. I never thought that our proteasome could do that.
Exactly. So questions like that, which are a super good question, I guess we don't quite know the answer to yet. But it's it's super fascinating.
That's amazing. And I mean, there's a lack of companies working on creating new antibiotics, just because in the industry there aren't that many incentives for companies to work on antibiotics because the returns are not as high. So a lot of these have to be government initiatives or government funded grants. Interesting. So this is a great way to kind of incentivize more companies to work on this.
Super interesting. Okay.
Wow.
Any other? Did we cover that?
I mean, I think that was a pretty groundbreaking March for 2025.
Oh yeah, 100%. Every month is like that.
Follow Sam on X or Twitter if you haven't already.
Awesome. That was fun.
That was amazing. Well, we'd love to know what you think. So please tell us if you would like to see more of these studies. Do you want more science? Do you want more medicine? And yeah, Sam and I will check the comments. Yeah.
Awesome. Thank you. Nice to see you, Tiff.
Nice to see you. We'll talk to you next time.